Bismillahirrahmanirrahim. Today the topic that needs to be explained is about mutation with a specific reference to the sickle cell anemia and Down syndrome. So first of all, we see that what is mutation? It is a sudden spontaneous changes in the structure of genes or in the chromosome number. Mutation can be inheritable, harmful and recessive, beneficial, that is, it arises in the nature, produces variations in the population, or it acts as a raw material for evolution. It means that mutations are random genetic changes. Now, when we consider the causes of a mutation, first of all, we see that mutation happens spontaneously and continuously, but their frequency can be increased by exposure to the following mutagenic agents. When we talk about mutagenic agents, they are those agents which brings about changes in the genes. So there are some physical factors or physical agents like some radiations like gamma rays, X-rays and ultraviolet rays, which can damage bonds and cause changes in the base sequences. Beside it, there are certain types of chemicals, for example tar in tobacco, which increases the rates of mutation and can cause cells to become cancerous. Beside it, the chemicals which are also causing mutation are mustard gas, formaldehyde, cyclamate, that is the artificial sweetener. Now, I will give a general consideration to sickle cell anemia and then we move on how gene mutation occur to bring about changes in the hemoglobin molecule. So, first of all, we see that what is sickle cell? It was the first genetic disease to be described in terms of gene mutation. A gene mutation is a change in the base sequence of DNA. We'll be doing in detail of that how this base sequence causes the changes in the DNA molecule later. But now we are going to consider that if there is a mutation in the hemoglobin molecule, then this causing the red blood cells to become stiff and sometimes sickle shape when they release oxygen to the body tissues. Now, here you see that this is a diagram of the normal RBCs, which is having biconcave layers, and it is having normal hemoglobin which can carry oxygen readily. And this is a sickle shaped RBCs which is having abnormal hemoglobin. Because of this abnormal hemoglobin, it is going to cause a sickling shape of this RBCs, and due to the sickle shaped RBCs, what does it do? That it causes the blocking of the blood vessels. So the blood is not going to flow smoothly through these blood vessels, and when it is not going smoothly through these blood vessels, it causes the tissues to not be supplied with oxygen and nutrients for its use. So due to the blocking of the blood flow, if it is occurring in the joints or bones, so the person is suffering from the painful crisis, and if it is occurring in the brain or eyes, like it causes the strokes, blindness or damage to the lungs, kidneys or the heart. Now here I'm going to explain you how the gene is mutation occurring in the hemoglobin molecule. Now this slide consists of two types of structures, like here we are talking about the normal hemoglobin and here we are talking about the abnormal hemoglobin, which is going to form this abnormal hemoglobin molecule. Now you all know that the DNA is having nitrogenous bases, that is adenine, thymine, guanine and cytosine. When they are going to line up, they form normal sequence of amino acid. When they join together, they form a proteins, and these proteins, that is the globin proteins, which combine with hemoglobin to form normal hemoglobin. Now, and the genotype of normal hemoglobin is HbA. In case of the mutation, if it is occurring, the mutation is occurring in the nitrogenous base pairs, then this mutant nitrogenous base pair, they line up and the amino acid sequence is being changed and when these changed amino acid sequence is going to join together to form a mutant protein which is causing the abnormal hemoglobin, that is HbS. Now this is the diagram which is showing that the base pairs in the normal hemoglobin causing the normal protein to be formed. That is when the base pairs is being transcribed and it is being translated normally, a normal protein is being formed. But if there is a mutation in the base pairs, so it is causing the mutation in the formation of the protein. That is initially in the normal hemoglobin, the globin molecule is having glutamic acid, which is a normal protein, and in case of mutation, this glutamic acid is being changed by valine, that is a mutant protein, and due to this it is causing the sickling of the cell. It means that if there is a 3 base pairs, it codes for 1 amino acids. So amino acid here, it is a glutamic acid in the normal protein and is the valine in the mutant protein. Now, how the sickling in RBCs occurs? The sickling occurs because of mutation in the hemoglobin gene. It begins with the substitution of valine for glutamic acid at the 6th position of the beta globin chain. This hemoglobin S aggregates, distorts RBCs and decreases the cell's flexibility. Now, if you can see that how this sickling is occurring, that if the faulty hemoglobin produces fibers inside RBCs, you can see the fibers inside the RBCs. The RBCs pull the fiber inward. The RBCs then become sickle, and due to the sickling of the RBCs, it is going to lodge into the blood vessels and it stops the blood to flow through the blood vessels freely. And then when it is stopping the blood vessels, it is causing the blocking, and the sickle cell crisis occurs at this stage. Now here we are talking about different hemoglobin genotypes, that is normal hemoglobin and the abnormal hemoglobin. Now in case of normal hemoglobin, the genotype of normal hemoglobin is HbA and is homozygous dominant, and in this case it is a normal allele, so the RBC structure is normal and it is carrying the oxygen normally to the tissues. And in this case, one is heterozygous, that is, it is having HbA/HbS. It means that some of the cells are normal, having normal hemoglobin, and some of the cells are having the sickled hemoglobin, that is, it is causing the sickling of the RBCs. So normal and both abnormal hemoglobin coexist in the RBCs, and it is a mutant allele. In this, it is not showing the symptoms unless the oxygen content is required in a higher amount, like in case of exercise and when the person is going at high altitude. So at that time, this symptoms of the heterozygous sickle cell anemia becomes prominent. Now in case of homozygous recessive, that is, the genotype of the homozygous recessive is HbS/HbS, and this is the mutant allele which is showing the gross symptoms because of the sickling of all the RBCs, and this sickling is going to hinder the oxygen carrying capacity of the RBCs. So in this case, the sickle cell crisis occurs and the person suffers from gross sickle cell symptoms.